Group 1 DNA Extraction Experiment. This is our group members. What is DNA? DNA tells an organism how to develop and function, and is so important that this complex compound is found in virtually every one of its cells. DNA, which is short for deoxyribonucleic acid, it is basically the blueprint for everything that happens inside an organism's cells. In this activity, you will make your own DNA extraction kit from household chemicals and use it to separate DNA from strawberries. Objectives: to know how to extract DNA from strawberries. Second, to observe what DNA looks like to the naked eye. Let's begin! Materials needed: strawberry, plastic bag, 2 teaspoons dish soap, 1/3 cup of water, 1/4 teaspoon salt, rubbing alcohol, and glass. It's procedure time! Step 1. Get ready with all the materials needed. Step 2. Put some strawberries into the plastic bag and seal it. Step 3. Squish the strawberries thoroughly in the plastic bag. Step 4. Make the extraction solution by mixing water, salt, and dish soap. Step 5. Pour the extraction solution into the plastic bag. Step 6. Seal the plastic bag again. Step 7. Press to extract more strawberries liquid. Step 8. Pour out strawberries liquid through a coffee filter into a glass. Step 9. Use a spoon to press on the residue that left over on the coffee filter. Step 10. Cover up the strawberry liquid and left it for 10 minutes. Step 11. Pour the cold rubbing alcohol on the top of the layer of the strawberries liquid. Take a look of the slimes that floating on the surface of the liquid. It's results time! The slimes are actually DNA of the strawberries. Let's watch a video discussion.
Let's experiment a little closer. DNA stands for deoxyribonucleic acid, and it's a molecule that encodes cells with the genetic instructions used in the development, division, and behavior of all known living organisms and even some viruses. Strawberries are octoploid, which means each cell contains 8 sets of DNA, unlike human cells which are diploid, meaning they contain 2 sets of DNA. We use strawberries in our experiment because of the large amount of DNA in each cell. Detergent molecules have 2 sides, a head which is attracted to polar water molecules, and a tail which hates water but likes to attach to non-polar molecules. Cells are made up of a phospholipid bilayer, with a polar phospho outside and a non-polar lipid interior. This property of cells makes them partially polar and non-polar. When our strawberries are squished and the cells are broken apart, the detergent tail attaches to the inner, non-polar side of the cell membrane and pulls the membrane pieces away into the water, effectively separating the cell membrane from the DNA inside the cell. This interaction between the soap and cells is much like how detergent will pull dirt and oil out of clothing. After the DNA has been separated from the cell membrane, the salt in our solution creates a conductive ionic environment which causes the strawberry DNA to bind and clump together. When we slowly mix the rubbing alcohol with our strained strawberry solution, we discover that DNA is not soluble in rubbing alcohol, which means they will not mix together. Because rubbing alcohol is less dense than our strawberry solution, it will layer on top of our strawberries, pulling the DNA away from our squished solution, allowing the DNA to form cloudy clumps.
Precaution steps. First, put rubbing alcohol into the fridge to help the DNA precipitate out of liquid. Second, the alcohol must not mix with the strawberry to ensure extraction process occur. Third, the alcohol concentration must be more than 70% of the solution so that the DNA extraction process will occur fast. Conclusion. The process of DNA extraction from the strawberries has been investigated. The DNA looks like slime when seen by naked eye.